Okay, great. So good afternoon, everybody. A very warm welcome to this webinar, which is the fifth in a series where we're exploring how the engineering biology mission hubs are harnessing engineering biology to address real-world challenges. So my name's Tom Pearson. I'm a senior portfolio manager for engineering biology at BBSRC. And in a few moments, I'll be handing over to the GlycoCell Hub team to take you through the really exciting work taking place across their hub consortium. But just before I hand it over to the team, I did want to spend a couple of minutes setting the scene, just in terms of why we consider engineering biology to be so important at UKRI, and also what we're hoping to achieve through these specific mission hub investments. Next slide, please, Vic. So, small bit of housekeeping to start with. So as this is a webinar, you'll note that your microphone is muted and your camera is turned off. We've also disabled the chat function just to ensure that all your questions are captured in the correct place. In terms of questions, you're very much encouraged to submit these to the Hub team throughout the webinar, and you can do this through the Q&A function. And then all the submitted questions will be collated and posed to the panel at the end of the webinar. And finally, we are recording today, and we plan to make the recording available on UKRI's YouTube channel sometime over the next few weeks, so please do keep an eye out for that. Next slide, please, Vic. Okie doke. So, engineering biology is one of our strategic priority areas for investment at UKRI. And the reason behind this is because we strongly believe that engineering biology has potential to deliver huge benefits to the UK in terms of addressing major societal challenges, driving economic growth, and also increasing national security, resilience, and preparedness. As an emerging technology, engineering biology has the potential to transform a broad range of diverse sectors, spanning agriculture and food systems, manufacturing and clean growth, in environmental monitoring and remediation, and also in biomedicine and in improving human health. And it's because of this broad range of applications that engineering biology is a technology that's really recognised globally to be of major importance, with some predicting that it could contribute alongside other biotechnologies up to around $4 trillion to the global economy over the next 10 or so years. Next slide, please, Vic. So, as part of our strategy to support the UK in realising engineering biology's potential, last year, UKRI invested over £100 million in 6 engineering biology mission hubs and in 22 engineering biology mission awards. These investments were made through UKRI's National Engineering Biology Programme, with co-funding support from the wider Technology Missions Fund programme, which has provided a substantial amount of investment to grow UK capabilities in critical technologies. The Mission Hubs and Mission Awards really build on the UK's world-class engineering biology capabilities to drive engineering biology to that next level of technology maturity, unlocking its potential across a broad range of application areas, and ultimately delivering tangible impacts that make a real difference to how this technology benefits UK society. So now I'm very pleased to introduce one of the Mission Hub project leads, Professor John Heap from the University of Nottingham, who, along with his colleagues joining us today, is going to explain how the GlycoCell Hub is harnessing engineering biology to transform the production of glycans and glycan-based therapeutics. Thanks very much, and over to you, John.
Thanks a lot, Tom. I trust everyone can hear me. So as he mentioned, I'm John Heap from the University of Nottingham, and we're pleased to have the opportunity today to introduce to you a little bit about GlycoCell, our UKRI Engineering Biology Mission Hub. Next slide, please. So this is an overview of the webinar. So this is what we plan to cover today. You've heard already from Tom about the UKRI angle, and then I'm going to, in a few moments, give you an introduction to GlycoCell at a fairly high level. And then the main event today is to hear from some of our brilliant early career researchers with some spotlight talks from them. And they cover some of their interests and the motivations around their work. It's not everybody or all the early career researchers involved in GlycoCell, so think of it as kind of a taster session to give you an overview and a flavour, but there's more to hear from us in future. Then after that, we'll hear from Victoria Wright, who is our hub manager, about our work toward achieving impacts. And then we'll wrap up with a little bit of good news we're keen to share with you about developing future leaders. And then we anticipate there should be time at the end for potentially for some questions and answers, which Tom will chair for us. So that's the plan. Okay, so we want to start with a few sort of big messages to sort of set the scene for our interest in our research and those of others working in biomedicine too. Next slide, please. So first of all, 1 in 8 deaths are linked to bacterial infections alone. So obviously that's a huge number, and this is not a new phenomenon. This is something that's been the case for a long time. Next slide. And vaccines work, and vaccines cut antibiotic use and antimicrobial resistance deaths. So there's a link here which people aren't always aware of between vaccines and vaccination and antibiotics and antimicrobial resistance. So to spell that out, taking vaccines decreases, stops people getting disease, and the absence of that disease means people don't need to take antibiotics, and there's a big decrease in prescription of antibiotics for that reason. And so there's less opportunity for those disease-causing bacteria to encounter antibiotics and become resistant to them. So this is really important and interesting link. And we're talking about the order of billions of doses of antibiotics and millions of deaths. Next slide. However, against the backdrop of those previous 2 messages, only 1 of the 6 bacteria most responsible for AMR deaths has a vaccine. So you can sort of see there the scale of the issue that represents. Next slide. So meanwhile, in another part of biomedicine, humanized biologic medicines are life-saving, but often unaffordable. And some examples on the right-hand side here. So this is referring to sort of modern biotechnology-driven medicines arising in recent decades that basically mimic human proteins to use them as medicines. So they're really impactful, but especially when they're new, very expensive and unaffordable. And obviously that significantly impacts access. Next slide. So what all those previous things have in common is glycans. And that brings us to GlycoCell's mission, which is safe, scalable, low-cost biomanufacturing, designer glycans and glycoproteins for vaccines, therapeutics, and diagnostics, or transforming biomanufacturing for health. So that's the big picture take-home message about GlycoCell's mission. Next slide. So let's unpack some of that. So first of all, what are glycans? Well, glycans are custom polymers using sugars as modular chemistry. So polymers being large chemicals, large molecules composed of building blocks or monomers put together in a particular way. And there are a lot of polymers that are important in biology and biotechnology and biomedicine. Some of them are much more well-known and more studied, like DNA or RNA or protein. And in a lot of those, they're much easier to study and easier to engineer, and we have well-established rules and frameworks for how you can specify with clear instructions which monomers should go together in which order to make a custom polymer. That's not really the case with glycans. They're much more complex and challenging, which we'll come to. Now, glycans are abundant on protein, cell, and virus surfaces. You could say they're sort of everywhere around us, but hiding in plain sight. So in that second image, the black dots are bacteria, and the pale light-colored halo or circle shape around each black dot is glycan. So that's this polymer, in this case, a capsule stuck to the outside of a bacterial cell. So there's a lot of this material all around us. Thirdly, they are critical across biomedicine, and we'll come to that point in a moment. And fourthly, they are challenging. We sometimes think of them as dark matter, by analogy to sort of dark matter in space, for biodesign. And that's because even though it's all around us, they're harder to study and harder to engineer. Next slide, please. So glycans are critical across biomedicine. So why is this? So across the top of this slide, we have several sort of well-known global phenomena that I expect are familiar. So this is antimicrobial resistance, emerging pathogens, climate change, growing and aging populations, and challenges and problems with the environment. And all these things together feed into infectious disease and non-infectious disease. And that's both the burden of those diseases today and what we anticipate in future. So starting on the left with infectious disease, bacterial and viral surfaces are glycosylated. And it's those surfaces that are key to immune recognition, because that's what the immune system encounters, is the surface of a bacterium or a virus. And that's why glycans are essential and needed for vaccines and diagnostics. You have to have the proper chemical structure of that surface, which is glycosylated, to have vaccines and diagnostics work well. Now, on the right-hand side of the slide, non-infectious disease. Well, over 85% of human proteins are glycosylated. And the glycans are key to the function of those proteins. So for that broad class of biomedicines called biologics, which involve basically copying or mimicking a human protein to use it as the basis for a therapeutic, well, the glycans are essential there to have effective and safe therapeutics. So you can see why glycans are critical across biomedicine. Next slide. So they're important, and we need to make them to use them for these different kinds of biomedicines. But there are real issues with existing routes to manufacturing. So this is why we are talking about replacing conventional synthesis with a safe, scalable approach. So the first row here is about conventional glycovaccine production. So this is conventionally done by growing a large amount of one of these hazardous pathogens, and immediately you can see the problem with that. And then a series of chemical steps are used. First, to harvest or collect the glycan from the surface of the pathogen and attach it to a so-called carrier protein to make the full medicine, and then to purify it. And these things together result in a range of sort of quality, purity, and cost problems that are linked together. And it means that ultimately these things are more costly than we would want and less suitable for low and middle income countries or for veterinary medicine. The second row is about conventional biologic medicine production. And this involves using specialist mammalian cell culture. So something that looks more like a research lab situation than what you would ideally want in a manufacturing setting. So it involves scientific expertise, and it's kind of fragile and poorly scalable as a production approach. So these things together mean that we have cost and scalability problems of this conventional approach. So the third and final row at the bottom, this is GlycoCell's vision, which is for both those kinds of medicines. We want to use safe microbial strains, which are engineered for production of glycovaccines and biologics, and we end up with low-cost, scalable, safe, responsive, and robust production. Next slide. So as I've touched on, glycans are challenging to study and to engineer. So that's why we built a multidisciplinary collaboration. And so the middle of the slide is the key point here, where we brought together the fields of experts from the fields of engineering biology and glycoengineering together, and that's GlycoCell. So this is the sort of engineering biology being the modern version, if you like, of biotechnology, which applies engineering principles, and glycoengineering, which emerges from the science, from glycoscience, and applying it to things that we talked about in biomedicine. There are lots of different approaches we're able to take with the scale of this consortium, many of which are shown around the outside, and I won't mention them now, but some of those come up in some of the talks you'll hear in a moment. Next slide. So GlycoCell is a UK-wide collaborative hub, and I'll emphasize we're collaborative. We're very keen to hear from people who could see a way or it would be interesting to explore working with us. And it brings together those engineering biologists and glycoengineers and glycobiologists I mentioned. There's 6 academic institutions shown on the slide and our 3 original core industrial partners. And then the investigators are named down the left-hand side, and that team has grown since the award with all the researchers and others shown on the right-hand side. And there's a couple more yet to come as well. Next slide, please. Okay, so to wrap up my introduction, I thought it'd be nice to share some examples of the ambitious sort of big goals we're able to take on with something, thanks to something the scale of the hub. And these are around scale, speed, and lean industrialization. So first on scale, we're building the capacity to so-called win the arms race against bacterial pathogens, by which we mean developing prototype vaccines against all the variants of a particular bacterial pathogen rather than just a couple of them. On speed, we're building the capacity to tackle a 100-day epidemic rapid response challenge. And on lean industrialization, we are building GlycoForge, which is a high throughput, low cost, and will ultimately be replicable prototype vaccine development facility. And that work substantially enables the 2 goals above on scale and speed. Next slide, please. I think that's my last slide, in fact. Yeah, thank you.
Hi, I'm Rebecca. I'm based at the University of Nottingham, and I'm going to give a tangible view from the bench on the early benefits of GlycoForge automation. Next slide, please. So GlycoForge is GlycoCell's focused biofoundry. It's this new platform that we've designed to do just one thing really well, which is to generate safe microbial strains which produce glycan-based medicines. But what does this mean in practice in the lab? Next slide, please. So first of all, why do we feel that we need to automate this process? Well, lots of genes are needed to make a glycan. So you need genes that make the building blocks of the glycan, and then you need further genes that assemble these building blocks together to make your full glycan structure. And we as a collective group are aiming to make lots of different glycans. So depending on your glycan target, there can be upwards of 20 genes involved in making just one glycan. And practically in the lab, this involves repetitive pipetting into one single tube for every single gene. And so as you can imagine, if you have 20 genes per glycan and you want to make lots of them, you're quickly going to become a very sad scientist surrounded in loads of tubes, and you'll have a very painful hand from your repetitive pipetting, hence our sad scientist here. And also this does take lots of time in the lab as well. So this is why we want to automate this process. Next slide, please. So in our GlycoForge, we're making use of some equipment called an acoustic liquid handler. So this mixes together tiny volumes, but really, really fast. So the equipment we're using is called an echo. So the echo uses sound waves to determine where the surface of a particular liquid is in your plate. And then it uses this information to release a really tiny 25 nanoliter droplet up into your reaction mixture plate. So in this way, we can give the echo a set of instructions that will mix together all of our liquids involved in making every single gene assembly, and it will do so very quickly and very accurately as well. So on the right-hand side, that should play in a second, you can see an example of a set of instructions that we might give the echo to make, in this case, 25 different gene assemblies. But the important point here is that all these individual colors that are popping up are an independent pipetting action, which means you can imagine this would become very repetitive very quickly. And hence, with this automation, we now have a happy scientist who doesn't have a painful hand. So that's good. Next slide, please. So where are we up to with this GlycoForge pipeline? Well, we've been able to build a prototype glycan pathway. So this is this 10-gene glycan pathway shown in the orange and yellow colors here. So we've been able to build this using the echo with high efficiency, and we've been able to generate a diverse expression library, hence as well shown in the orange and yellow colors of the genes. And in this way, it means we're able to select the fittest clones of E. coli that not only produce lots of our target glycan, but are also happy to do so as well. So this pipeline is now in use in the lab for lots of our target glycan-based medicines. Next slide, please. So this example shows how GlycoForge development is already making an impact on our ability to generate prototype glycan-based medicines. Thank you.
Hiya, I'm Mark Harrison, and I'm based at the London School of Hygiene and Tropical Medicine. And what I'm going to present to you today has been done in quite close collaboration with our GlycoCell partners at the University of Dundee. And so I'm going to be talking to you a bit about glycoconjugate vaccines for neglected streptococcal diseases. Next slide, please. So glycoconjugate vaccines are proven for Streptococcus pneumoniae, but there are none licensed for other streptococcal diseases. There are 500 million cases of group A streptococcus every year, which result in 500,000 deaths, while Streptococcus ubris causes cattle mastitis and is a major economic burden globally. Next slide, please. So the aim of this arm of GlycoCell is to deliver low-cost glycoconjugate vaccine candidates against important streptococcal pathogens. And as the first two that we've chosen, we've gone for a major human pathogen, which is group A strep, and a major veterinary pathogen in Streptococcus ubris. And the reason that we work with glycoconjugates for vaccines is that they make really great vaccines because you get a very strong immune response that lasts a long time. So glycoconjugates are formed of a glycan coupled to an immunogenic carrier protein. And the reason they work really well is because of this combination. If you give a vaccine which is just the glycan, which comes from your pathogen of interest, then you get quite a weak immune response that doesn't last very long. Hence why we couple the two components together. But as John alluded to, some of the production methods that are used for producing our glycoconjugates, they're very expensive due to their kind of complex nature as a result of the chemical reactions often being multi-step and therefore requiring multiple quality control steps as well. So this makes the vaccines unaffordable for significant portions of the world. So we are looking at using an alternative, which is bioconjugation. So in bioconjugation, we use a safe host organism, such as E. coli. And into our E. coli, we put genes that encode our glycan, the carrier protein, and the third component, which is an enzyme which couples the two components together. And as part of GlycoCell, the aim is to integrate some novel engineering biology techniques in order to improve our success rate with bioconjugation and also increase the amount of glycoconjugate that comes from our host organism. Next slide, please. So how are we getting on so far? The project's gone really well so far. We've been able to demonstrate production of 3 novel vaccine candidates against Group A strep. So that means we've successfully conjugated our glycan of interest from Group A strep onto 3 different carrier proteins. And in doing so, we've been able to show that by integrating these novel engineering biology techniques, that we've been able to significantly improve vaccine production in comparison to existing bioconjugation technology. And so the next step for us is that we're moving into scaling up production of one of our vaccine candidates. So when we demonstrate production of vaccine candidates, these tend to be in cultures in the milliliter range. But now we're looking at pushing that up into the litre range, because that'll give us a good idea of how much vaccine we're able to produce from our host organisms. And in doing so, we can look to optimize production methods to further increase that vaccine yield, as well as produce enough material in order to test and validate our vaccine candidates in animals. In terms of Streptococcus aureus, work is well underway on expressing the glycan from Streptococcus aureus in E. coli, and we are going to follow a very similar path to Group A strep, in that we'll then look to conjugate this glycan onto carrier proteins and then scale up production. Next slide, please. So in summary, this is how GlycoCell is making an impact on delivering vaccines for neglected streptococcal diseases.
Hello, I'm Ryan Griffiths and based at the Quadram Institute in Norwich, and I will briefly introduce a snapshot to our approach at building an advantageous Bacillus platform for glycan biomanufacturing. Next, please. I want to just start by highlighting the many World Health Organization recognized gram-positive bacterial threats mentioned today, such as Streptococcus pneumoniae, Group B streptococci, but also highlight antibiotic-resistant Staphylococcus aureus strains. And what is critical to this work is that they produce extracellular O-glycosylated adhesins. And these adhesins are really important for the bacteria to attach to host cells and surfaces to form biofilms or aggregates. And another aspect is that they are encoded in both clinical isolates and non-typable isolates and have become a target in vaccine development against these gram-positive bacteria. Next, please. And so there are several reasons we decided to use Bacillus subtilis as a host for the production of some of the O-glycoconjugate targets, or expressing these large pathogenic O-glycosylated adhesins. Subtilis is also a gram-positive bacteria, and there are some advantages in keeping these things consistent between gram-positive origin organism and a gram-positive heterologous host. Another aspect is that Bacillus subtilis has been optimized for genetic amenability and bioindustry scalability. And so I'd also like to bring in some attention to the designation as a safe host for the production of a whole range of biomolecules. Several strains have been granted qualified presumption of safety by the European Food Safety Authority, the equivalent being generally recognized as safe by the FDA in the US. So therapeutics such as proinsulin, vitamins, the enzymes in your fabric softener, and carbohydrate polymers like hyaluronic acid, destined for food and cosmetic use. These are some of the things produced and manufactured in subtilis. So another key feature is the ability to develop subtilis as a carrier and producer of vaccine and adjuvant technologies. And several of these are in preclinical and clinical trials for humans, and some are moving towards real-world deployment for animals. Next, please. So in our lab, we have further engineered Bacillus subtilis to produce O-glycosylated glycoproteins. This is originally based on our research into the secretion of glycoprotein adhesins in the gut symbiont Lactobacillus reuteri. And here you can see on this cartoon model the sequential cytoplasmic attachment of the glycan by specific glycosyl transferase enzymes. And this glycoprotein is then regulated and chaperoned by other dedicated proteins to the cell membrane, where it is translocated by a specific translocon complex. And the movement across the cell wall is then carried out by some of the endogenous enzymes present in subtilis. And ultimately, our glycoproteins are found in the extracellular medium. Next, please. We have genetically modified subtilis of these secretion pathways so that our new strains can secrete glycosylated adhesins. I mentioned earlier present in some gun symbionts, as well as from the pathogenic streptococci. And this really opens the door to recapitulate some of these O-glycosylated adhesins from more clinically relevant strain or disease-causing strains and take advantage of the synthetic biology competence in this hub to address challenges in yield, scaling, or vaccine development, as well as be amenable to the high-throughput engineering biology approaches such as the GlycoForge. Next, please. And this is how GlycoCell is reimagining Bacillus as a platform for glycoconjugate biomanufacturing and vaccine delivery.
Hello, I'm Stuart, working at the University of Nottingham, and I'm going to talk to you today about GlycoCell's work to develop synthetic genomic mimics of fungal pathogen glycans for biomedicines. Next slide, please. So, the monosylation of cell surface proteins drives fungal immunogenicity, yet studying these glycans has been limited by technical barriers. By engineering Saccharomyces cerevisiae to mimic pathogen glycans, we can unlock high-throughput tools for glycovaccine innovation for target fungal pathogens. Next slide, please. So, why target fungal pathogens? Well, invasive fungal diseases are an under-recognized emerging global health threat that is estimated to affect millions of people worldwide. The global burden of these fungal infections is growing, with an estimated annual incidence of about 3.8 million deaths and 6.5 million serious fungal infections every year. As such, the World Health Organization has identified Candida albicans and Aspergillus fumigatus as a critical priority, based on factors such as average case vitality, number of cases annually, complications, and antifungal resistance. Fungal infections with a particularly high mortality rate include invasive aspergillosis infections of the lungs, with about 1.8 million deaths per year, and candidemia of the bloodstream with about 1 million deaths per year. However, yeast infections of the mouth or vagina are more common and are usually less serious. Next slide, please. So, what can we do about it? Well, we aim to mimic the glycan structures that drive immunogenicity in these fungal pathogens by using safer, easier to manipulate, and optimizable model organisms such as the budding yeast Saccharomyces cerevisiae, which some of you might have at home for baking or for brewing. So we aim to identify the genes responsible for these immunogenic glycans within pathogens, extract, modify, and synthesize these genes, and build the pathway in a safer model organism such as Saccharomyces. The result would be a safe, yet easily modifiable Saccharomyces cerevisiae cell, which would express pathogenic glycans and therefore appear to the immune system as if it were a pathogen. We could then exploit the properties of such a system for future learning, or more importantly, translate this into candidate glycovaccines. Next slide, please. As an example of this, we've been able to identify a set of genes of interest from Candida albicans which are not present in Saccharomyces cerevisiae. And it's thought that these genes are thought to be important for the differences in immunogenicity between these two species. We have subsequently designed, synthesized, and assembled this gene pathway using Golden Gate assembly and transformed it into Saccharomyces cerevisiae. We've been able to identify through flow cytometry cell sorting and through next generation sequencing Candidate Saccharomyces cerevisiae cells, which express these foreign Candida albicans genes, enabling them to mimic the mannan structure of Candida albicans. We will now be able to characterize this candidate, explore variants, and ultimately develop it for its future use and properties as a therapeutic. Next slide, please. This is how GlycoCell is engineering safe cells to mimic fungal pathogens for vaccines and biomedicines. Thank you.
Hello, my name is Tej and I'm based at Imperial College London. Today I'm going to be talking to you about brewing therapeutic glycoproteins with conditional humanization. Next slide, please. So yeast produces around 20% of approved biologics, offering fast and low-cost production, ideal for low-resource settings. However, yeast has high manostructures, which differs from human glycosylation. This causes a key challenge for therapeutic production. Next slide, please. So you'll be able to see 3 glycan structures on your screen. The leftmost glycan structure is the native glycosylation machinery for yeast, and this is our yeast starting point. You'll be able to see that this is hypermanosylated, so these are the glycans shown in the green circles. But what we would like to make is more human-like or complex type glycans shown in the glycan structure on the rightmost part of the screen. This structure has glycans such as galactose shown in the yellow circles and N-acetyl glucosamine, for example, in the blue squares, which are more complex glycans. A key milestone for my project is actually to be able to produce a MAN5 structure, which is the glycan structure in the middle, which only has 5 mannose residues. So this contributes to a lower number of mannose residues compared to the yeast starting point, but also gives rise to a biantennary structure that then allows us to build more complex glycans to obtain human-like or complex glycosylation. But a key challenge in being able to achieve this is that hypermanosylated proteins is integral for membrane physiology in yeast. This is shown in the green cloud around the yeast on the left-hand side of the screen. The current practice in industry is actually to knock out an enzyme called OCH1. This is a mannose transferase. This adds more mannose residues to existing mannose residues that gives rise to these hyperbranch structures. But knocking this protein out means that there's a consequence. There's altered physiology, which leads to clumping of the cells, and this eventually leads to problems to do with scalability and biomass limitations. So what are we going to do about it? The next slide, please. So currently, I'm working on building 3 different switches that allows us to switch between the hypermanosylated and non-manosylated state. An example of a toggle switch that we're developing is something that's inspired from the bacterial toggle switch design with 2 arms expressing repressor proteins that mutually inhibit each other. An example of an autoinducible switch is shown in the middle, where the carbon source itself can be used as an inducer or a repressor molecule for synthesis or depletion of our protein of interest. And the last strategy is actually based on a plant-based design where a protein of interest can be tagged with an auxin-inducible degron. And upon the addition of auxin, we're able to selectively degrade the protein of interest. And in this case, the protein of interest could be the OCH1 protein that hypermanosylates our therapeutic protein of interest. So all of these 3 switches would be able to allow us to switch between a hypermanosylated state that makes the yeast happy into a lower manosylated state that allows us to produce more human-like glycans. Next slide, please. So in summary, 3 novel genetic switches are being designed and validated for production of humanized therapeutic glycoproteins from yeast. Thank you.
Hi, so my name is Victoria and I'm the GlycoCell Hub Manager, and today I'm going to talk to you about how GlycoCell achieves impacts. GlycoCell is building a broad community of experts to translate our science into real-world impacts. We will deliver transformative biomedicines, transformative capabilities, and develop future leaders. So let's have a look at how GlycoCell is forging a community. GlycoCell is a collaborative hub and we're always seeking new collaborations. All of our members join our monthly all-hands meetings where we discuss our progress in our research projects. We encourage our ECRs, that's our early career researchers, to work together, and we've created an ECR-only forum to enable this. We've already created cross-hub publications, and we also hold in-person events, and these include our annual all-hands in-person meetings. We have strong and active relationships with our 3 core industrial partners. And for example, we've already transferred some of our science to one of these, Iceni Glycoscience, who are currently industrialising that for us. As a team, we regularly present at international conferences. We take part in panels which highlight the path of from translation to translating our research. We have hosted accelerator roadshows, and we're incredibly proud to have hosted a ministerial visit. GlycoCell is focused on translating our research. And this is really important to us, but you can imagine that turning a research product into a commercial product takes a great deal of money, specialist know-how, and specialist equipment. So let's take a look at some of the things that we've put in place to enable this. Members of the GlycoCell team are part of existing vaccine hubs, which have been set up by the government or funded by the government to address the challenges surrounding vaccine development. We have strong and established links with IP specialists, including IP specialists within our particular field. One of our core partners, Inceptor, has significant experience in the global biomanufacture of vaccines, and we've had in-person meetings with these and have agreements in place. And as you've heard from Mark, the GlycoCell team has already transferred one vaccine prototype to one of our partners for scale-up. GlycoCell is incredibly proud of our early career researchers and technical professionals. These will be the future drivers of innovation. So it's really important that we put things in place to help them with their careers. One thing that we've done already to help towards this is we have trained 14 GlycoCell members in advanced design of experiment software. And this has been done by our core industrial partner, Synthase. We're also developing a bespoke training programme for our researchers, which will help them build specialist knowledge, bridging engineering biology and glycoscience. And we're also going to ensure that we provide the right amount of mentorship and training and commercialisation and entrepreneurship to help them in their careers. I hope these slides have given you an insight into some of the things that GlycoCell is working on towards delivering impact. I'm now going to hand over to John for the final slides. Thank you.
Thank you, Vic. Excellent. Yes, so as Vic has mentioned, developing people and developing future leaders is really important to us. So it's nice that we have a few items of good news to share with you on that front. So from left to right on this slide, first of all, Margarita Spencer, who was one of our first GlycoCell postdocs here at Nottingham, recently successfully secured an assistant professor post at the University of Warwick. So that's really exciting. She took that up recently and is now able to start building up her own lab. Next in the middle, Tej, who you heard from a few moments ago, who's one of our existing GlycoCell postdocs at Imperial. She recently successfully secured a fellowship at NUS, which is in Singapore, and University of Edinburgh, and she'll be taking that up next year. So that's excellent. And I'm super excited to be able to share with you, we're announcing today that Dr. Rochelle Orr from Stanford University is joining GlycoCell in Nottingham as an assistant professor very soon, in fact, in November. So these are 3 great examples of developing future leaders, and I'm sure there'll be lots of exciting stuff to see from these guys in the future. So very much a case of watch this space for them. Next slide, please. Great. So really just that brings us to the end of the sort of main part of our what we wanted to share with you today. So a few words of thanks. Obviously, thank you to everybody watching for your attention. And Tom, Ashley, UKRI, and everybody else there for arranging and hosting the webinar, as well as funding the hubs, of course. And then GlycoCell people on the call. So the early career researchers, Rebekah, Mark, Ryan, Stuart, and Tej you heard from, hub manager Victoria. And we also have online on the call a couple of the GlycoCell investigators, Ben Blount and Alex Falls-Payne, have also worked on this webinar with the support of the rest of the consortium. So we have time, I think, for some more questions in a moment. But before that, in case that isn't the right format in which you want to contact us, we really would encourage you either way to please reach out. Our LinkedIn is on the screen via that QR code on the left, and you can email us. It's written on the screen, and there's also a QR code on the right there. So please don't hesitate. If after this, if ideas sink in and you want to reach out, we'd love to hear from you. So thanks very much. I think it's back to you, Tom, now.
Excellent. Thanks very much, John, and the rest of the GlycoCell Hub joining us today. So we do have some time for questions now. So please do submit those through the Q&A function. I can't see any in there at the moment, so perhaps I can kick off with one. So it's really great to hear the kind of close links that you have with the sort of original industrial partners that you had within your grant, and especially that you're already actually transferring tech through those. But I just wondered whether you could kind of provide your view on what other aspects of your research program might be particularly interesting to potential new industrial partners.
Yeah, I'm happy to start on that one. Others might have comments as well. So I think yeah, we are super open to engaging, across the piece really. And I think there's an element to which we don't know what we don't know. So there might be people out there with ideas and ways to connect that we haven't considered. So we really would hate to be prescriptive and put anybody off. So I think the answer is broad. I think the existing core industrial partners, they are the core partners for a very good reason, and that's sort of illustrative, which is Synthase and Iceni have they're leaders in what they do. They're specialists in technical areas that support the technical research mission of the hub in very specific and helpful ways. So Iceni is a specialist in glycoscience, and Synthase is an expert in design of experiments and robotic automation and distributed experimentation as well. So those are sort of engaged with that. So by example, if there were others who thought there was a mutual benefit, or we could help them, or they could help us in terms of how the technical research is delivered, that would be one example of an excellent sort of scope for an opportunity there. And then the other partner, Inceptor, they are a vaccine and biologic manufacturer, right? So they do the scale up. They develop things themselves, they scale them themselves, and they manufacture them. So that's important for us because, as Vic mentioned, the total costs involved and the time and work involved in taking a prototype vaccine and getting it all the way through to impact to market and to patients is a big endeavor, well beyond the scope of what any sort of small collection of people could do on their own. So partnering is critical for that, and they are one of our partners, but we would love to develop more such partners, and we have happily, as Vic again mentioned, because we have existing members of GlycoCell who aren't actually on this webinar right now, but that's Brendan Venn at LSHTM and Karen Polizzi at Imperial, who work with ECR colleagues who are on the call. So those guys are members of the 2 existing vaccine hubs, and those do provide us with a network that's established specifically to sort of solve that same problem of what is the translation pipeline. So there may be people who are part of those, or recognize that sort of formula of needing to collaborate to get things through to translation, that would be another sort of scope where I imagine it would be a really good fit for a new partner.
Great, thanks very much, John. So still waiting for other questions to come in. Perhaps just pause for a moment. Ashley?
Yeah, thank you, Tom, and thank you so much to GlycoCell. That was a wonderful presentation. Yeah, my question was around thinking of the research that you're conducting at the moment, the considerations around responsible research and innovation, which is something I know that we think a lot about in research as a whole, and particularly around the hubs too, and the work that you're doing. So I just wondered, going forwards as well, with into the translation as well, with the application of your research, the work and considerations that you're thinking as a consortia there around responsible research and innovation.
Ben or Alex, do either of you want to pick that up first? I mean, it would be ideal if we had Karen on the call for some of that. She often speaks to this piece, but like, Ben, do you want to say anything on that?
I do. I mean, I think we try to bake responsible research and innovation into everything we do, rather than just being a side consideration at every point during the research process, and also, trying to scope out exactly how we're going to translate this. We try and make it a core part of our decision-making process. We're really lucky as well that in the consortium, we have members who unfortunately aren't here, who will probably answer this question a lot better than me, who have, they have these prior experiences of rolling out actual vaccine products, often to, basically to underserved areas of the world. And they have some great learnings from that in rolling out vaccines and other such products into communities where maybe there are issues around cultural sensitivities towards that uptake and general attitudes towards these products. So we're really lucky to have members of the consortium who have experience with this, have really good success stories, but also challenges that they can feed back to all of us, and just help us keep this in mind through every stage of the development.
Thank you. That's really good to hear as well. And it's great that it's, like you said, it's embedded within the programme of work as well. That's fantastic. Thank you.
And a couple of things I could add to that are, in some ways, some of that is easier for us than it is for some other advanced biological technologies, because although the technologies we're using are new, the idea of a vaccine isn't a new concept, or a therapeutic that's intended for human use isn't a novel concept. So the sort of regulatory pathways are clear, and what it means to show a medicine is safe before giving it to humans or to animals for that matter is understood. So we don't have to sort of reinvent the wheel on that part. And another part of it, which you probably recognize from across everyone who's spoken today, is, how we pick our targets. So we could work on any disease. You could pick a disease that only affects people in a certain part of the world or a certain type of person, or you could pick diseases to target that are, have very large numbers of people or smaller number of people. And all those are part of the RRI agenda, I would argue. And I think you've heard from a lot of people today sort of those rationale. Some of these are diseases that affect large numbers of people. And so that's part of the RRI piece as well.
Thank you, John. Yeah, there's definitely going to be a huge impact in the work that you're doing as well in the research. And like you said, you can clearly see that those are considerations that have been embedded in the work that you're doing too. So that's wonderful to hear. Thank you.
Thank you very much. So we have a question here from Gavin. And this is a question for Ryan specifically. So Gavin says, nice method. Have you made the O-glycosylated secreted SRAP? And what do you intend to use it for?
We have not made that particular SRP yet. Using these different O-glycosylated adhesion secretion pathways, we chose a panel to test at first the range of capacity for how many can be translated over. And then part of these ones could go through into developing glycovaccines against targets, but also this kind of secretion pathway is to be developed into a platform. So to take advantage of specific signal peptides, build truncated versions, novel, different types of conjugates that maybe have better immunogenicity, things like that. So we're at the beginning of this project. But the perspective is strong that this kind of pathway is translatable into Septilis in this manner.
Thank you very much, Ryan. Pause for a few more seconds just to see whether any more questions come in. But I guess just to reiterate again, if anyone attending the webinar today or watching back on YouTube wants to get in touch with the Hub to discuss your work, how would you go about doing that?
Vic, do you want to mention?
Yep, so on we have a LinkedIn site. So if you go to LinkedIn and search for GlycoCell, that should come up. And also we showed the QR code on the webinar before. And we have an email address which you can contact us on, which is glycocell@nottingham.ac.uk. Thank you.
Great. Thanks very much, Vic. Okay, well, I think we can call it there. So just falls to me to thank the GlycoCell team again for a really interesting and insightful webinar. We will be holding another webinar sometime in the future with the Preventing Plastic Pollutions Mission Hub, led by Andy Pickford at Portsmouth. Please keep an eye on the UKRI events page for that. Thank you very much, everybody. I'm happy to draw that to a close now. Thank you.